Okay, good afternoon. We are very pleased to have Professor Hsiu-Hau here with us today. Okay, Hsiu-Hau received his bachelor degree from our department in 1991, and he got his PhD degree okay, in 1998 okay, in Santa Barbara, UC Santa Barbara, and postdoc in Indiana University in 1999. Then went back to National Tsinghua University in 2000, and he is now a professor in the physics department of Tsinghua University. Hsiu-Hau is very well known for his work in condensed matter theory, okay, especially in nanoscale spintronics, okay, and statistical mechanics. He received a couple of work from National Science, Ministry of Science and Technology. In addition to be a very good researcher, he is also very well known for his teaching. Okay, his course, in the open courseware, okay, received as award twice, which is, I guess, is unique in Taiwan, Taiwanese in the world. Okay, in the world. Now, recently he extended his interest, okay, to quantum biology. Okay, that's his topic in the past few years. So today he will give us a talk on the dawn of quantum biology. Okay, I'm really very eager to know what is that. Okay, cool. So let's welcome him.
Thank you. So it's nice to come back here. So I guess everyone can hear me. So this is going to be a rather controversial subject. So if you do not agree, feel free just to interrupt me, and I'll try to debate or fight back or then provide more evidences. The last time that I was invited to a workshop at KITP in Santa Barbara, and this is a bunch of biologists. They're all outstanding researchers in old fashion, that smell. And they seem to enjoy attacking a theorist. Yeah, that's me. And I have been spending more than 3 weeks, and I don't really think I did a good job. And I hope that after coming back to physics department, most of you got some background trainings and probably has a little bit more of quantum mechanics. Maybe we can have some common ground to appreciate what happens here. So this is a fairly new subject. So basically, since this is a colloquium, so I'm going to make it relatively simple. So first, I'm going to talk about what is quantum biology and what kind of example you expect that it might happen. Because as you know that in the abstract, usually in biology, everything is wet, and temperatures, room temperatures, and so you don't really think that quantum coherence will ever survive. So why would we be interested in quantum mechanics in biology? Okay, but if this is really what you think, then we can probably think twice, because nowadays there are emerging experimental evidences that this is actually not so. And at some microscopic scale of biological processes might heavily rely on quantum mechanics. So let's wait and see. So in order to convince you, I will probably first introduce about 20-year pursuits of the photosynthesis and how quantum coherence was after long times of experimental and theoretical pursuits, and it was found at ambient temperature, not at some crazy wire low temperature, at ambient temperatures. Then I will talk about the main subjects, the olfactory receptor. Oops, sorry. That is the main subject here, is how we smell. So, and then I will give you some theory and prediction, and I will assume that most of you are trained in physics department and thus probably have zero common sense in biology, and I will try to be as methodological as I can be. So, but if somehow my statement or this technical jargon confuse you, I can translate them into the ordinary terms that physicists can understand. So the first one is really this beautiful, nice review articles appear in Nature Physics. If you're interested, I think you can find most of the pieces in this paper. And one of the authors, Chen Yunan, is actually a professor at National Chengkong University. So as you can see, the review article has a very elegant, strong, and clear punchline, that is quantum biology. So the key word is that can we really find quantum coherence in biology or not? So they have actually provided several examples. The first one is actually photosynthesis, that is this one. Okay, and there are also radical pair magnetoreceptions. And then after these two major examples, there are olfaction, vision, long-range electron transfer, and enzyme catalysis. Okay, so I'm not going to go over all these 6 topics. So I will focus on the photosynthesis and olfaction in this talk. And for those of you who might be interested in the radical pair, it's basically that how does avian birds navigate themselves? You know, they travel a long distance, so they don't have GPS, they don't have iPhone either, so how do they really keep their directions? And there have been really puzzling things, and now it seems that they can actually detect the gradient of magnetic field. Not gradient, not magnetic field. Because if you just detect magnetic field, you can easily get confused by artificial wiring and all sorts of things. But then if you're detecting how the magnetic gradients, you can actually rule out lots of artificial noises. So this is actually one of the very interesting things. And the photosynthesis is probably the most well-known things that we now have confidence to say that we indeed find some quantum coherence. So here is photosynthesis. Okay, so let me try to introduce you to how things happen. So, photosynthesis is basically that you need to transfer the energy carried by a photon eventually to some chemical energy which can be used by living beings. So how do you do that? Well, it's a very complicated chain of reaction. Here, even though this figure looks very messy, this is the first step of photosynthesis. So basically, a photon comes in and then hit the chromosomes complex. And then the energy basically hop around and eventually will transfer to this, what we call FMO. FMO stands for 3 people's name. And then somehow the energy will propagate along these FMO proteins and find what we call the reaction center and then transfer the photon energy downwards. So it's all about energy transfer. And usually in biochemistry, you might think that what happened is that this energy just diffused from here to here and then to here. But amazingly, according to experimental finding, the efficiency seems to be much higher than just random diffusion. So then, basically, the physicists strongly suspect that maybe some sort of transfer is actually done by quantum dynamics. So we will focus on this FMO protein. So you can sort of think about this as the second layer of energy transfer, and you have a bunch of FMO forming a network, since it's like hopping. So if it's really quantum mechanical, it should be relatively simple. It's just like a one-dimensional, two-dimensional hopping chain and hopping network. And you can actually calculate the energy transfer efficiency, and it will be actually larger. So if you zoom in the FMO, okay, it's still not simple. You will find that FMO is a trimmer. So what does a trimmer mean? Trimmer basically means that they are almost identical 3 structures like this, connected together and make an FMO. So remember, this little tiny FMO here has this very complicated structures. And each monomer here has 7 pigments. So 7 pigments are basically 7 large molecules. So in total, you basically have 21 pigments. And so how are you going to study this? Well, let's just focus on just the 7 pigments for a moment. So if you assume that there are quantum hopping between them, you can actually solve this Hamiltonian. And then you will get 1 to 7 energy levels like this. You can actually calculate the transfer rate between them, either within the Lindblad dynamics, or you can use higher calculations, and you can find a major pathway of this. The question is, how do you know that transition is quantum mechanical or classical? So here is the idea. So particularly for students, suppose now I have a black box. This black box is so complicated, and there is a sound fault inside with resonant frequency omega 1. How would you know that you have some sound folk inside. So usually what we do is that we just get it a kick. And then if the output has some sinusoidal oscillations, then we know there is a sound folk inside, right? So can we do similar things, basically? So this 7 pigment networks is very complicated. So the only way we can be accessible is basically just like try to detect this invisible sound folk by stimulations. Okay. So the usual simulation is that we just kick it once and we collect the response. It turns out that this is not smart enough. Why? Well, suppose now in the box we have 7 energy levels, right? So that means there are tons of transition frequencies. So you have basically collections of sound folk inside, like this one. So one way to do that is that you can now scan, you have an input, which is, let's say, omega tau, and you scan this omega tau and see whether you have a resonant output or not. And if you plot it up, then you will find when the omega tau hits the resonant frequency, omega 1 and omega 2, you have large signal. So now try to plot this together. If I use omega tau, the probe, as my horizontal axis, and the response, omega t, as the vertical axis, then you should expect when you're moving around this and plotting all your data like this, there should be 2 huge spots like that. Okay, so that's the way how you actually use laser light and optical means to basically try to deposit the energy positions in these pigments. But then so this is experimental data. It's apparently more complicated, right? So this was actually published in 2005. So you can see, yes, they are diagonal terms. So from this diagonal spot, you can actually try to inverse calculating what happens and what are the energy level. And it turns out that you can actually calculate this to the 5th order of perturbations and it matches with theoretical prediction rather well. But then something more. You find these off-diagonal terms show up. Okay. So I'm not going to zoom into the whole detail of how you can actually get this response contour. I'm just saying basically that if you have sound folk with omega 1 and omega 2, you use omega tau to probe it, right? You should only have diagonal peak. That is that when you hit the frequency omega 1 or you hit the frequency omega 2, you should receive very large responses. But here you found sometimes an interesting thing happens. That is when omega tau is hitting omega 1, but somehow you are receiving omega 2. So it's almost like now you're trying to sing in C and then you're hearing A. Well, that's something amazing. So what happens? Well, it's not that surprising. You can sort of imagine now if somehow you couple these 2 folks together. So that's why that when you have an input like this and when you have output like that, you will have off-diagonal terms, right? Because these sound folks are coupled together. Now let's try to translate that into what happens in this biological system. What happened here is relatively simple actually. That is, what happens is that because of this setup, in experiments, what they do is that they shoot in the first kick to excite the system. And this will actually produce an entangled state between 2 excitons. That is, you actually have an entangled state between 2 energy levels. And then the second kick do the probing, and the third kick read out the signal. So this is really sophisticated, what we call the 3 pulse pump probe experiment. Usually pump probe experiment is only 2 pulses. So here you need to do 3 to really see the off-diagonal term. And why is that so important? Because in order to show that this is indeed quantum coherent, you need the off-diagonal term to show the oscillations. And you can see the experimental data for 77 cases. You see these very beautiful oscillations. And this solid line is a predictions basically from the theoretical calculation. And if you hit it up to 180 Kelvin, it's still there. So I hope that a series of work like this actually extend more than 15 years. That basically a group of theorists and physicists believe that there should be some quantum coherence in photosynthesis. So So what they do is they cool down these whole things and then try to find it, and then gradually crank up the temperature.
Excuse me, what's the energy of all this peak? I want to know the scale.
Oh, you mean this oscillation scale?
Yeah.
This oscillation scale is from, I think it's around, oh, here. It's femtosecond, so it's really, really fast. And that's why that even at this temperature you can have them. So how to estimate this, or I hope that after I providing this, that you can erase one common confusion. That is that usually we think that you need to lower to low temperature so that you can see quantum coherence. And that's actually not necessary, because the world is ruled by quantum mechanics. It's all about time scale. So even at room temperature, you can ask yourself, at what time scale that you can see quantum coherence? And it turns out that in this particular case, if you have a time scale which is like 100 of femtoseconds, that's short enough. So that in fact you can just see these quantum coherence there. So thermal fluctuation is actually not the major problem to make it coherent. It's more about the biological environment. It's whether the biological environment will provide additional fluctuation, usually that would kill the quantum fluctuations. So it's not just the random vibration that you expect in molecules. And that's also the things that usually, for instance, while I was working on condensed matter physics, we have one of the biggest conference called low temperature, LT, right? And why? Well, because in condensed matter systems, you need to go into the low temperature to see the quantum coherence. But here you don't need to do that. Yeah, since the connection is a bit lagged, so let me switch the remote. So give me 30 seconds. Sorry about this.
So that means that
yes.
So your quantum coherence exists
Mm.
only for a very, very short period of time.
Exactly. But if that's only it takes for a biological process to happen, then you will find out. So see, the trick is so simple. It's whether you believe it or not. And so usually our biological responses are really slow. For instance, in neuron transport, you can actually use a very classical circuit to stimulate most of the fantastic brain activity and things like that. But then what about detections? So one can actually think about what is photosynthesis. Photosynthesis is actually a sensitive detection of photons. So it's just like high-energy particles, particle physics, that how you capture this whole thing. And that process can be really, really short. And if you just look into the biological textbooks, you will find that all this happen in cartoon way. They just say that, well, a photon comes in, and it got absorbed by reaction center, and what's in between. So you don't really see any significant biological description about this, and that's why that in recent years, this process has been basically put under the spotlight and have been re-examined.
But whether it has an effect or not will depends on the response time of the detector, right? But can the detector respond so quickly?
Yes, that's how you see, right? So you can actually see that how this reaction center received the whole thing is actually through the quantum ripples. Otherwise, what does this mean? What this means is basically that if now I'm showing something on this and you receive the response, if it's complete classical diffusion, then you will see this will just decay, right? But if somehow this is propagating like wave-like, then you will have riddles. And since this riddle appear in the off-diagonal peak, which is directly associated with the off-diagonal quantum entanglement, so it got to be quantum wave, the quantum riddles. And that's why that people have designed this from the simple kick to palm probe, and then to 3 ports palm probe, and finally convince the community that that is the case. So that's a very good question then. That's why it takes a very long time. But I guess it's good for them, because they're good whenever they got results like this, and you just found that the technique is getting better and better, the temperature is getting higher and higher, but then most of the publication always appear in Nature of Science. Because they are the only crazy guys who believe that this can be done. Yeah. So finally, so let me just skip all this complicated detail. And one important thing is that you basically need to probe the entangled state to ensure that the ripple you see is not classical waves, it's quantum mechanical waves. And so the key point of this whole thing is that in 2010, about 6 years ago, this is done at ambient temperature. So you have found this. Basically, that when you shine light on this, and you actually found quantum ripple propagating through the reaction center. Well, that's pretty amazing. So that's a very short introduction to this huge photosynthesis stories. And I completely skipped the theoretical part because I'm a theorist, and I know that if I want to introduce something like this, and I just quoted lots of theoretical paper, that would make it very suspicious. So that's why I apologize if you are serious and have been working on this. I mentioned zero amounts of theoretical work. But trust me, most of the experimental work published in Nature and Science, so that make the theoretical work actually appear mostly in Nature Physics. So it's kind of funny. But the good old time has ended about 2 years ago. Yeah. So now let's switch topics. So okay, that's one thing. That if the biological being want to detect photons, and it will have some quantum coherence. What about smell? So let's really go to the fundamental questions. How do we smell? If you think about it, this is truly amazing. When we smell, we inhale a bunch of nanoscale molecules. And somehow your nose, miraculously, can differentiate these molecules. And if you're doing matter experiment, you know how hard it is. This is extremely hard, even nowadays. But somehow everyone got this nanomolecule detectors, and we seem to abuse them. So we didn't quite appreciate that. So how do we really smell? So even before then, before 1991, most people even believe that when we try to smell, some of the chemical molecule might diffuse into our body. So be really careful because you can get poisoned by that. But then luckily, that's not what happens. So let's just go through this and give you some of the biological sense about the smell. So the first one is we do have olfactory receptors. That's the only receptor which is sneaking out the skin. All receptors in our bodies is inside our body, okay? But then this is the only one. So this was found by Linda Buck and Richard Axers back in 1991. So they actually won the Nobel Prize for it. So this is a pretty interesting thing, and making the puzzle even more puzzling. That means that we have detectors to detect these molecules. So you might ask, so how many kinds of receptors we have here, right? Apparently there's an important question. Well, so let me just give you a brief rundown on how you smell. So if you are interested in more detail, you can basically just read this amiable. I found it an easy reading review articles in Nature back in 2001. So the first one is this. Okay, the order molecule actually bind to the receptor like this. So basically in the cilia, we have lots of receptors evenly distributed, and the molecule somehow dock on these receptors. The second one is that the olfactory receptors are activated, so they are neurons, and then the neuron signal will then rewire here, and the signal will be relayed in glomerulus. Glomerulus is basically, if you got confused about the glomerulus, basically it's a neural node, like a ball. And why do we need that? Well, because the receptor signal can be very random, and you want to increase the signal to noise ratio. So you just relay most of the light signal together to enhance the signal. And the last one is this, of course. just like we joke about. So, yeah, you don't really need your sensories, right? You only need your brain to feel the joy. And so the final processing in the higher region of the brain. So there are 4 processes. And I think most biologists believe that none of these processes involve quantum mechanical things. Here, I'm going to only target the most boring ones.